welcome to another segment of The Harvest. On today's episode, I'll be interviewing Mr. John Climaco, CEO of CNS Pharmaceuticals on the NASDAQ exchange with the ticker CNSP. John, welcome.
Hi, Carmel. Great to be here.
Very excited to have you. We are thrilled to hear more about the most recent news. So as a start, please highlight what CNS Pharmaceuticals does, leaning please to the latest news with this in-license of the late-stage novel potential blood-brain barrier. But I'm going to leave that all up to you because I'm sure you can explain this in much better detail than I can. Please go ahead.
Sure. So we are a oncology drug development company. Specifically, our space is neuro-oncology. So that's cancers of the brain and the central nervous system. So traditionally, these are some of the most difficult and deadly cancers, very challenging to treat because they lie beyond the blood-brain barrier, which is this very specialized network of cells that protects the most important organ in the body. And so it's very hard to get drugs and treatments to the brain when you have tumors growing in it. So that is really what we specialize in. We have a very exciting compound called berubicin. It's a first-in-class drug that penetrates the blood-brain barrier and treats brain cancer. This is in a late stage designed to be pivotal trial with over 250 patients in the United States and in Europe currently enrolled in the study. It's fully enrolled. It'll be reading top line data in the first half of next year. So we're very excited about that. We've been working on that since the company's inception back in 2017. But the news that you mentioned is equally exciting after many years of searching and diligence and evaluating over 200 different compounds for potential in-license. We settled on an amazing drug called TPI-287, which is a novel abiotaxane of the taxane family. And this is also a unique blood-brain barrier penetrant drug that was specifically designed to treat these very difficult brain cancers, such as glioblastoma. So this drug has already been used in 350 patients in a series of phase 1 studies, and we have scooped it up, and we are now just at the beginning stage. We just announced this yesterday, so it's hot off the presses, and we are just getting started on planning what will be another designed to be pivotal study for this very exciting drug. So in the last 24 hours, we've doubled our pipeline, and we're very excited about that.
So, berubicin seems to be your initial lead candidate, and highly exciting news with the new candidate in place. But what sets berubicin and, of course, now this the new one apart from anything else in development out there for glioblastoma?
Yeah, awesome question. And it's really at the heart of what we do. We are a bit of a contrarian play in neuro-oncology and frankly in oncology in general. What you read about in oncology today are a lot of very sexy immune-based therapeutics that we refer to them basically as rifle shots because they're really each designed for a specific patient. They've had some spectacular results. However, they have almost uniformly failed to produce durable results in CNS cancers. And so the need here remains very distinct. There are about 50,000 patients each year in the 8 major markets around the world that develop glioblastoma, which is the most aggressive and deadly primary form of brain cancer. And as I said earlier, the problem is that this cancer develops inside the blood-brain barrier. And so this specialized network of cells and molecular level pumps keeps otherwise helpful drugs from getting to the site of the tumor. And so what makes us different and what makes our drugs different is that rather than taking a rifle shot approach, what we said, and I'm a big believer in personalized medicine. I spent a decade in genomic medicine. I love it. I think we're still waiting for the promise. But in this space in particular, what we don't have is that one-size-fits-all cytotoxic chemotherapy solution to go into the armamentarium to help these patients recover time and quality of life and let them live longer. You have to understand, these patients, when they get diagnosed with glioblastoma, they have on average 9 to 12 months to live. So it's a very aggressive cancer. And on recurrence, that's after they've failed first-line therapy. And first-line therapy in this disease is surgical resection, followed by radiation and treatment with a drug called temozolomide. But temozolomide is has sort of two unusual features. Number one, it's the only drug approved for primary brain cancer treatment anywhere in the world. And number two, it only works in about 4 out of 10 cases, because the other 6 out of 10 of us will have a genetic anomaly that renders that drug totally ineffective. So for 6 out of 10 cases, if you can believe this, in 2024, we have no chemotherapy option whatsoever for these patients. It's a really disastrous scenario. And I often say it's the great, non-discriminate cancer. It doesn't matter if you're Ted Kennedy. It doesn't matter if you're John McCain. It doesn't matter if you're Beau Biden, the son of the president. It doesn't matter actually if you're the president of the MD Anderson Cancer Center, where berubicin, our lead drug, was developed. If you get this disease, you are going to die. The best treatments in the world today are almost totally ineffective. And so what we said was, rather than aim for a rifle shot, what if we took a little different approach, turned the telescope around, so to speak, and said, okay, what if we took a class of drugs? In these two cases, we took a class of drugs called anthracyclines, which are among the most useful and well understood and characterized chemotherapy drugs in the world. They've been around for 60 years in clinical treatment. And in the case of our new molecule, the general class of taxanes, again, which have been around for decades and are revolutionary when they were developed. And we know that those two classes of drugs destroy glioblastoma cells in the test tube all day long. You can do it very easily. There's nothing magic about it. People have been doing it for decades. The problem is when you put them into human beings, the blood-brain barrier defeats most of the drugs in those classes. So what we said was, what if we could modify these molecules to bypass the blood-brain barrier? If we could get these drugs that we already know are efficacious against these tumors into the blood, inside the brain, what would happen then? And what we've seen are some really spectacular results. So I think what separates us from others in general in this space is we have the advantage of starting with molecules from a class that we know are effective against these tumors. The issue is the transport. And in both cases, in berubicin and in our new drug TPI 287, these very significant, well-studied, well-understood, safe molecules were modified in each case to make them what's called highly lipophilic. That is to say, they have a very strong attraction to fat. And our brain is the most fat-rich environment in our body. And so these molecules are so attracted to that fat-rich environment in our brain that they can actually bypass this very powerful passive and active network of specialized cells that comprises our blood-brain barrier. And once these cells get in the brain, and we have seen this activity in previous studies for both drugs, we see some really incredible results. We start to see patients behaving much like patients whose cancers are declining. And that's really what we want to see. So we've got two molecules. These are basic cytotoxic chemotherapy. Everyone else is sort of paying attention to immuno-oncology, which is great. But we're paying attention to the foundation that is still missing, the one-size-fits-all treatment that can extend what we call both progression-free survival, that's stable disease, and overall survival, which is just what it sounds like. And the key measure, patients living longer than they would otherwise without these drugs.
Thank you. Thank you. That's an excellent response and really informative. But also you highlight that this potential for global reach, the study that was with berubicin, it sounds incredible. Can you speak more about that study in a little more detail? So tell us about your patients and what you saw in terms of efficacy. And then also, once that's complete, tell us about the next milestones that we can look forward to.
Sure. So berubicin was studied in a very nice phase 1 several years ago. And that was just a dose escalation study to identify the maximum tolerated dose. But what's interesting when you study drugs in glioblastoma is this. This cancer never spontaneously resolves. That is to say, if you get this cancer and it's left untreated, you will die. Sadly, even if it is treated, you will die. You'll just die a little bit later today than you would otherwise. But if we see that trajectory change, we know that we are seeing treatment effect of whatever drug or treatment or therapeutic was introduced into the equation. So in the case of berubicin, when it was studied in the phase 1, these were very sick patients, very heavily pretreated patients, dozens of rounds of chemotherapy, the type of folks that typically don't respond any longer, and sadly are probably weeks away from death. We saw more than half of those patients achieve stable disease. We saw 3 patients out of 27 see their tumors shrink. And we saw one gentleman whose tumor completely disappeared and remains cancer-free many, many years after that study was conducted. So those are the kind of results that when you see them, you know this drug is having an effect. Similarly with TPI 287, studied in over 350 patients in various phase 1 studies, but in the phase 1 study looking at patients with glioblastoma, we saw a 60% overall response rate. We saw a 40% of patients stable at 6 months, and most importantly, overall survival was 13.4 months. Now compare that for 4 to 6 months for patients who are untreated at recurrence. Again, these are the patients who failed first-line therapy. Their cancer has now recurred. Data tells us that their median survival is between 4 to 6 months. With TPI 287, those patients were living a median of 13.4 months. So a massive improvement in overall survival, literally, nearly a year of additional life for these patients. And so we are very, very excited about both drugs and their potential to go forward. And those are the kind of data that, when you see in this disease, you say, we have something here, because most drugs, first of all, most drugs simply fail to get into the brain in any kind of therapeutic concentration. And then when many drugs get into the brain, those few that do, most of them are defeated by the cancer itself, because this is such an aggressive and dangerous cancer.
Makes total sense. Given, in the hopes of assuming completion of successful clinical trials and with obvious needed regulatory approvals, what would you say is the potential market size for berubicin and the new drug that you just mentioned?
Yeah, great question. So we have a really good idea about that. So I mentioned this drug temozolomide. So temozolomide is a classic alkylating agent chemotherapy drug, been around for a long time. It's a first-line therapy. So basically after radiation, you get temozolomide. What's interesting, again, only 4 out of 10 cases will respond to temozolomide. That means 6 out of 10 get no benefit. And in most of the world, frankly, really anywhere where you have centralized, single payer type healthcare, they really don't use temozolomide for those, what we call them unmethylated patients. These are the patients that won't respond. So remarkably, for these patients with this terrible disease, in 2024, they literally have no chemotherapy option whatsoever. They go from radiation to hospice. It's a very, very chilling thing to understand about the population that we're working on. But that drug, for as poor a performer as it is, and being off patent for over 15 years, is a $9 billion a year drug globally. And it's almost exclusively used in this space because of its ability to penetrate the blood-brain barriers. So, the analog that I speak of is we don't really know exactly what the market will be for one of these 2 drugs, but we certainly know that it begins with a B. And we can tell ourselves that if a drug that only works in 4 out of 10 cases generates $9 billion a year in revenue as an off-patent drug, we think there's a very, very healthy market for these drugs that actually do materially extend overall survival for these patients.
In light of all of what you've just mentioned, how is CNS Pharmaceuticals positioned financially to advance clinical programs, particularly in light of recent funding activities?
Sure. So, it's been a very big challenge. I'm a contrarian by nature, so I don't mind doing something that is different from what the rest of the field is doing. I think there's great rationale for what we're doing. The downside of that is that, we have not ever really drawn the attention of the market in a way that I think would be commensurate with what the opportunity is that we have. That said, we have been fortunate among microcap biotechs right now to would be able to finance. We have steadily kept the financing going. So, we reported minimal cash at our last quarterly report, but we have done some raises that we've announced since then. And so I think we're fairly well positioned now. We will have to raise a bit more capital to finish our berubicin trial, which is aimed again for the first half of next year. And then on the TPI 287 front, I want to make sure that I say that, one of the attractive things to us and what made us, I think, the best suited company in the world to develop this particular drug is, again, we're very specialized in the sense that we're not just looking at any therapeutic options. We're looking at traditional cytotoxic chemotherapy options. And so we're very skilled at both developing those drugs and running them through clinical trials. We have an excellent CMC team that has really done incredible work on berubicin and a commercial pathway. They'll be doing more of the same with TPI 287. But importantly, we have developed a tremendous network of some of the leading KOL neuro-oncologists around the world, both here in the United States and in 7 countries in Europe. We have 45 centers currently participating in our global berubicin study. And our ability to repurpose that network for another trial with TPI 287, I think, is going to be a key feature of us keeping costs down and accelerating the timeline for the program. By that, I mean, what people don't often think about in clinical trials is that it can take years to enroll a trial of this size, particularly with an orphan disease, where in the United States, we only have 15,000 cases a year of glioblastoma. And we don't look at all of those. We only look at the ones that recur, and we only look at a very specific set of those that we feel are going to be the best responders here. So your numbers can get very small. It can be very challenging. It takes years to sift through which centers and which individual neuro-oncologists have a high enough throughput of patients and are excellent scientists, clinicians, to help us do what we need to do. We've done all that work now. All of that work can be repurposed. And that, I think, gives us a huge leg up in terms of what we need to do to get going on TPI 287. And also, I believe, will reduce the amount of cash that we will need to spend executing that trial for our new molecule.
Wonderful. So you've mentioned a few things that are really intriguing. Firstly, it's the lack of patience to really understand, firstly, the market size and also to do the clinical trials. So with that, what are the strategies or the potential to strategize expanding the market beyond glioblastoma, for berubicin's applications?
Yeah, so that's an excellent question. So if we think about what it is that we're doing, we are specialized in cytotoxic chemotherapy, the kind of traditional oncology, one size fits all or one size fits most proposition. What that does is it focuses on drugs that are, in this case, pretty known quantities, whether it's an anthracycline in the case of berubicin or an amiotaxane in the case of TPI 287. These come from classes of drugs that have been around for decades. They've been well studied. Their side effects and toxicities are well understood. So we know an awful lot about these drugs, and we also know an awful lot about how they work. So berubicin is what's called a topoisomerase 2 inhibitor. The shortest way to describe it is it's basically when a cell divides, you have to unwind a double strand of DNA and separate that DNA. Topoisomerase 2 is the enzyme that sort of cleaves the DNA apart. Berubicin disrupts that function and inhibits the cell's ability to divide. Similarly, over on the TPI 287 side, it's a microtubule depolymerization inhibitor. And what that basically means is microtubules are these protein strands that form the shell of a cell, if you will, and they move components of the cell around as the cell lives and divides. So one of the things that the cell has to do is go through a process called mitosis, which is when the nuclei separates into 2 cells. Well, a microtubule like TPI 287 disrupts that process. It prevents those microtubules from depolymerizing or shortening. They can't move the DNA around. And if they can't move the DNA around, there's no separation. The DNA doesn't move into 2 separate nuclei, and the cell doesn't divide, and eventually it dies. Similar ideas in both, but very different approaches. You can imagine what we're looking at are molecules that are intended to disrupt the ability of cancer cells to rapidly divide, which is what cancer cells are in the business of doing. So if you can disrupt that by a mechanism that is not unique to a particular type of cancer, and these mechanisms, the mitotic mechanism and the mechanism of TPI, or excuse me, of topoisomerase 2 are not unique to these cancers. There's lots of other cancers that are sensitive to disruption of these mechanisms. You could look at, for example, triple negative breast cancer, very sensitive to anthracycline therapy in the breast. However, this is a cancer among dozens that have a well-known pathway of metastases to the brain. And if you have breast cancer and you could treat it in the breast, that's great. If it metastasizes to the brain, sadly, you now have the same problem that the GBM patients have. You have a tumor growing inside the blood-brain barrier. We already know that those tumors are sensitive to anthracycline therapy because they work in the breast tissue. The problem with every other anthracycline except berubicin is they are defeated by the blood-brain barrier. Berubicin, our drug, passes through the blood-brain barrier. And once it's there, again, I like stacking the deck in our favor. When it encounters a triple negative breast cancer cell that is metastasized to the brain, we know it's going to have treatment effect on it because we know from decades that those cancer cells are already sensitive to the exact class of drugs that we're working on. The trick wasn't the mechanism. The trick was getting the mechanism to the right place.
Wonderful and so articulately explained. Thank you. I completely understand exactly what you're saying. And no, it's fantastic because, one has these... We have these preconceived ideas and we don't understand the intricacies. And I think what CNS Pharmaceuticals is offering is an extension, right? Needed. There's a gap, and you're actually making it, firstly, articulating such a great way for understanding, but also providing exactly what you're saying, that's connecting those dots and providing the treatments people need.
It's the reason I love what we do and I love this company because, it's great to be the smartest guy in the room if you are, but it's also great to recognize that, hey, maybe there's another way to do this. Maybe we actually don't need a completely novel mechanism of action. Maybe we can take something that we know already works and repurpose it. And what I often say is what we're doing is both with anthracyclines and with taxanes, these are the hammers in the oncologist toolbox. They're the tools that they most often use, they most often reach for. The problem in CNS cancers is the nail is located in the brain where these hammers can't go. We can deliver the hammer to the nail. And when we see the results that we see, it's not totally unexpected to us because we know these cells are sensitive to these type of therapeutics.
Excellent. Moving on to academic institutions and other biotech companies, do you collaborate with any of these to advance your research?
We have excellent collaborations. As I said, we have 45 centers around the United States and in 7 countries in Europe. These centers include some of the best institutions in neuro-oncology and definitely some of the leading KOLs in the space. These are the folks whose names are on the top of papers and are at the podium and all the society events around the world, who are really at the forefront of what we're doing. And they love what we are doing because they know, as treating clinicians, that while the excitement and the research dollars and the headlines in the general press go to some of the rifle shot amazing things that are being done in immuno-oncology, the bottom line is that the bulk of patients are still missing that foundational care. It's just not there in this space. And as a clinician, when you're actually, boots on the ground treating sick people, that's what you want. You want a therapeutic that you can give not just to one person that's going to cost, $1 million or $2 per treatment at a very specialized medical center. You want a treatment that, when your patient is in, city XYZ that doesn't have a fancy neuro-oncology center, but can be given a drug that is safe and effective and can advance their overall survival. That's really the tool that you're looking for. And that's what we are working on. And I think that's why we have so many of these academic collaborators around the world. And that's also why, frankly, we have enrolled our berubicin study as rapidly as we did. 252 patients, so folks understand. We don't know if it's the largest glioblastoma drug study going on in the world today, but if it isn't, it certainly is one of them. There's no question about that. It's an enormous number of patients. It really looks not coincidentally like a phase 3 study because we designed it and powered it to be pivotal. And the key is enrollment, is getting those physicians and their patients into the study. Folks thought that when we started doing this, it was going to take us 6 years to enroll a study of this size. We did it in 2.5 years. And I think the reason we did it is as fast as we did is, number one, we have a great drug. Number two, we chose excellent physicians who have really highly regarded academic practices, who know how to conduct great trials. And number three, I think those physicians, knowing that our drug was an anthracycline, and in the case of TPI, I think going forward, knowing that drug is a taxane, as a treating physician gives them some incredible confidence to deploy it and recommend our trial to these patients. Because remember, it sometimes gets lost that the bottom line for all of this kind of research takes place in an exam room with a very, very sick patient and their family. These folks have probably just received the worst news they've ever received in their life. They are trying to figure out what to do, and they are often astonished that there is so little that can be offered them. And the physician has to guide them through this process. And really, the only opportunities are clinical trials. And when you're choosing from the hundreds of glioblastoma trials that are out there, that physician, probably given the life expectancy of these patients, has one shot. They're not going to get to keep going. They're going to have one chance to make a difference for this patient. And so many of them have chosen our drug because I think they know, while it is not approved, while it is still an experimental being tested therapy, because this drug and because our new drug come from such tried and true, well understood, decades old classes of drugs, they can make that recommendation confidently. And those collaborations have really advanced our program tremendously.
About a month ago, we saw a reverse stock split, just to lean towards the shares now. And can you speak to us about that and how you see that reverse split, benefiting shareholders in the long term? Obviously, you're trying to increase the share value as many companies in your position do. But just speak to us and our audience investors out there listening about that.
Sure. Obviously not something that anybody wants to do, ourselves included. We went through a lot of analysis about what was the best course of action. The reality was that, we had a NASDAQ bid price deficiency that can't go on forever. And so, our listing on NASDAQ is one of the key assets in the company that we just couldn't put at risk any longer. And, when we did the analysis, kind of counterintuitively, it told us that companies that had been facially cautious in terms of the reverse split ratio tended to suffer the most after the reverse split. And those that put the most distance between a dollar bid price and where they came out, after their reverse split did the best. So we chose a relatively high ratio and we went for it. We all feel the pain as insiders in the same ratio that everybody else does. But what we have to do as managers of this company is keep our eyes on the prize, which is maintain our ability to finance the company, because without that, we can't proceed with our work. And our work and the completion of it is the only way that we create value on a lasting basis.
Great. And then in terms of resources, you mentioned, the most important part is the management, making sure that the company's cash runway is managed, etc. So what are your strategies going forward to address those?
Yeah, so, we are constantly on the road. My CFO, Chris Downs, and I talking with institutional investors, family offices, hedge funds, partners, large retail players, money managers. We are literally doing this every single week of the year, just got back from a long round the world trip, hitting major markets in Southeast Asia, Asia, and Europe, and telling the story of the company. And some of these things are long lead time sell cycles, but we think that that's the best use of our time to make sure that people understand the story, they understand fundamentally what we are doing, because at the end of the day, we firmly believe this company has been deeply undervalued almost since the IPO. And so the share price does not tell the story of the value that we have. And so we have to get out and tell that story. And if there's any doubt about that, I would say, look back at our IPO when, if we're being frank, we were really an idea. We hadn't started executing this trial at all. We had a drug and we had a plan, but that's all we had. And we were worth nearly 100 million. Now, fast forward, we're worth a fraction of that, and we have a trial that's nearing completion, and we have a second drug. We're not in a popular space. Biotech has not been popular. Chemotherapy is not popular, and GBM is a space that has killed not just patients, but dozens of companies. And so I think the bias is that the same thing will happen to us. But we think, as I've said now for the entirety of the interview, we have a different approach that I think is going to, bear fruit in the very near future.
Looking forward to it. And then let us touch quickly on FDA's grant of fast track designation and what that'll mean for, impacting Berubicin development time.
Yeah, a critical piece. So, I heard once from a former, director of the FDA, there's really only two rules to drug development. The first rule is figure out what the FDA wants. And then the second rule is just, for God's sakes, give it to them. We're not in the business of trying to outsmart the FDA. And the best way to figure out what they want is to talk to them. Unfortunately, they will never just draw you a roadmap and say, we want this, 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 and this. And if you do all these things, we'll guarantee that you'll have an approval. But you can develop a dialogue with them whereby you can understand the nuances of the numbers and where they're leading us. And it was clear from the beginning with Berubicin that where they were leading us based on the earlier data in this promising drug was a pivotal trial. They strongly suggested that we do the trial of the size that we did because they strongly suggested overall survival as the endpoint, which is the only endpoint, by the way, that they will accept for registration. So we have been aimed at registration from the day we enrolled the first patient in this study. And that's a little bit different process, I think, that a lot of companies go through. We took a bigger gamble, if you will, but we all said collectively, we know this drug works. The FDA appears to know this drug works, and they want it proved. So let's prove it, and let's do exactly what they said. And I can tell you we're going to do exactly the same thing with TPI 287. The trial design may look a little bit different. There may be more or less patients, but the idea of using FDA to tell us what to do is going to be the same.
Excellent. And that's a wonderful close to this interview. Thank you so much, Mr. John Climaco. NASDAQ ticker CNSP. This is CNS Pharmaceuticals, Inc. I'm Carmel Fisher. Stay tuned for more.
The Harvest is a production of Harvest Communications. Find it anywhere you listen to podcasts.